So today I will do the representation of the cell switches model, applying the Markov chain stochastic model, a check on between the two population with regards to two specific cases. One is the mRNA and the proteins, and the second is the neuron model. And for the second one, there are also some introduction of some concepts from quantum computation as well. So basically, the switching model here is derived from the predator and the prey type 2 function model, and there are the mechanism of the normals and the persisters. And the normals are with the intrinsic rate of increase, and the persisters are the instantaneous discovery rate and converting coefficient. And so the switching between the status here mainly can be applied on quite many models. So for the first one, in the, for example, the cell regulatory networks or the signal response on the excitability and inhibition. And also, like our first one is the mRNA and the proteins, and also the interaction of DNA and associated proteins. And in this paper is the HEX1 and the HS1 cell. The computation method here we use for the first model is based on the two concepts mainly. First is the diffusion case with the LDT, and the second is the mean time to switching. And let's see how we use it here. So the status of the switching mainly is the molecular interactions on the phenotypic data of the mRNA and its associated protein, and especially the trajectory of the production of that HEX1 and HS1 with the interaction with each other. And as I mentioned in the previous page, we use the least action method combining the diffusion process. And mainly it's in our computation, it's the diffusion process is refers to the concentration of the cell from high to low. And here the least action here is based on the Lagrangian method. And after we get to the Lagrangian here, we are trying to get the final formula of our Hamiltonian. And then in our calculation, the main tricks using is to find the 3 convergence points this is the very important step as the stability analysis. We can find actually 3 convergence points through the we use this method MST to get the 3 eigenvalue of the position quantities through satisfying h equals 0 and the Hamiltonian here equals 0. Then for the second method, it's also our switching model based on the normal differential equation. Similar as the first one, we have the attractors also on the eigenvalue computed method, and the attractors are found at the bifurcation points in single unit. This is actually just for the single unit method, and we use also the condition equations to set the equation equals to 0 and get the result. So how we get the equations are based on 2 important variables here. One is the membrane potential S, and the second is its phase. And here is this stands for the interaction in 1 unit, not in one, generally. And then it's based on the time. Then we go to so here is the total computation with the approximation numeric later. So the switching model, as I mentioned before, is basically actually it's the switch of two status, and if in a single population, it's like how we predict usually with label, like for example, it's labeled as 1, and another status labeled as -1, and there is the bidirection interaction there. For then it can goes to the second status here, and we see this first one as the unit, and this as the second unit. Similarly, for our neuron model, it's based on the inhibitory populations and the excitatory populations. And here, the first 1 unit here is from task negative neural network. And the second unit here is the task positive neural network. So it's quite similar, as you can see here. The population is for inhibitory cells. They're of the GABA, and for both these two. And if it goes to the excitatory cells, for the negative task, it's the NMDA, and for the positive task, it's the AMPA. And there are some more introduction on the diffusion method. And mainly it's like we saw on the solution of the diffusion process, we start from the bistability of the two-state model and get the initial condition and identification of the mean transition rate with the principal eigenvalue, and then get our approximation function. And using the least action method I said before, then we set the mean time optimization and get our Hamiltonian. So then if we go specifically on our model of the mRNA and the protein, this is I didn't write here, but it's based on also one classical model of the mRNA, the DNA research. And it's actually, this is already simplified from some parameters fixed as one here. And this is the diffusion model based on the degradation rate. And actually there is also, we introduce one dimer here. This is the a new variable we introduce here, but later can be cancelled. And so mainly it's based on the Poisson process, and we can here is actually the derivation I mentioned, how we set some parameters, and so after that we can get our Hamiltonian here. And as I also mentioned before, the actually this is the condition to find the solution of the Hamiltonian. So then later we try to randomize with the stochastic model here. And this is how the semantic probabilistic uncertainty conditional network. It's the same thing as this, actually just it's quite similar, just the same thing as the process of how it transfer from the switch of the hex 1, then to the mRNA, then to the protein. And if it cross to interact with the HS1 part, it first goes to interact with the DNA of the HS1 first, and then it again down to the mRNA to the protein, and the back response to the hex 1. So for the second model, it's then based on the membrane potential and the phase. For this model, we also try to get the normal form here from the bifurcation point first. And then there is on our data, which is based on that inhibitory and excitatory neurons. This is the one in one single unit. Then it is the either, for example, in the negative task, neural network task, then with the firing rate model and the leakage integrating differential. And the leakage actually is just based on the the Hodgkin model, which is quite like the origin of this. So then then I try to introduce some quantum computation as well to compare with the classically computed neural model. Actually, why I did this is because there are quite many similarities between the neural and the quantum bits, like how they finally normalize into the two states, and also like some properties as like all versus none. It either goes to all or goes to none. So for the quantum computation here, our main actually basically all computation are based on the Hamiltonian as well. And so you should, if completely computed, there needs to be the external and internal interactions. And but the external part is based on dipole-dipole interaction, and and the photon kick, which is usually at least here is not our focus. So we are focused on the internal interactions based on mainly with the phase gates operations, and its dynamics can be introduced by this standard Hamiltonian. This R state and about some quantum computation. This is actually just a method to note like one status. If in the more general ways, we write something like the 1 and the 0 is like more known to more used. So for this one here, what important is the Rabi frequency. It describes the exciting lasers, and gamma is the decay from the excited states. And the similarity between it and the quantum and the classical computation here is also The qubit goes from the ground state to the excitation state. Just like our neurons, we decide whether it spikes or not with the threshold. For quantum, we know if it gets enough energy, and it will go to the higher level. So it's really quite similar. And then there is the transfer of the Hamiltonian, which is also related to the phase. And for the computation of the phase, or for the computation of entanglement, this one is, there are actually two methods. This is the more selectively used one, and it's also can be quite intuitively get here, like if we introduce one more status here and get it to transport from the either 0 or 1 status, and we through those different transfer of the phase, we finally get it transported. And then there is the stability and analysis I have mentioned before. So this is and the main user method, like I said, is the eigenvalue method. Here, if you're interested, this is we just to get the fx and the gx settled, and through the partial differentiation, we get the matrix here, and with that, with all the conditions equals to 0, then we compute the result of the eigenvalues. And then we substitute it back to our Hamiltonian. Then we get the fixed points. It is the same thing for this neuron switching model as well, but based on the membrane potential and the phase. So, and this is the stability analysis of it. And through the final results of it, we can see how it's transferred from the, we say, the persistent and the and the normals. Because we use the Hamiltonian method, what we are interested in is also the dynamics. The Hamiltonian for classical computing is related to the position and the dynamics of the P of the quantity. And then this is the how we get the transfer matrix. So there can be also some related analysis done later based on our results. Like most directly can be seen as the residue and the phase transfer. For our neural model here is also like here is the results of the bistatus. Like I said, find the stability, the stable points. And in the quantum mechanical, here is how to express it with the also eigenvalue. Here, this is the eigenvalue, and the then goes to the Hamiltonian in quantum computation way, and try to find the collisional phase. So generally, and just I mean the switching model is mainly like because here I just use the bi-state test, so it's the two between the two population. There are either only interaction of one direction or the two directions. How it switch from the first to the second, either with the cross interaction or not. So and this is the I'm glad to have the representatives today, and this is my supervisors in the East China University of Science and Technology, and we are from the Dynamic Neural Lab. And thank you.